Hi, my name is Dr. Tal Altman. I'm the CEO of SynVaccine, which presents the breakthrough in rational design of viral-based products. Viruses aren't just negative, as we tend to think of them, but they are nature-made vehicles that can be utilized in many ways. They deliver DNA to specific cells, fix faulty genes, help defend the body as in vaccines, and recently have been recognized by the FDA as a safe and effective therapy in the fight against cancer. But none of this is helpful if we can't find the right virus with the right traits for the job at hand. The problem is that development of viral-based products still works like it did in the past, relying mostly on random trial and error. It's a slow, expensive process, full of safety concerns. And in the end, it gives us zero control over the virus we do end up using. That's no good. Sophisticated modern vaccines, let alone oncolytic viruses, need to have extremely specific traits. For example, oncolytic virus need to replicate fast in the tumor, be visible to the immune system, but not too fast and not too visible, or be destroyed by the immune system. Vaccines need to look like the wild type, be mutation resistant, but otherwise be as weak as possible. You get the point. A virus that specific is extremely hard to achieve, unless you design it yourself. What SynVaccine does is develop customized viral-based products using its own proprietary platform technology that works in 3 stages of design, build, and test. So first we design. We analyze big data of viral genome, identifying and comparing patterns of millions of viruses throughout the evolution. Through the study and processing of data such as ribosomal movements, mRNA levels, RNA folding, methylation, among many others, we were able to formulate the very rules that dictate the traits and behavior of viruses. Based on those rules and our specification, we designed the virus. Then, we build our products using cutting-edge synthetic technology tools, putting computer code to life. Finally, we test our viruses in relevant biological settings. Breaking from current technology, our process is not a one-way, meaning that if we find out, based on the results, that some alteration is required, we can just go back to the design and do what any change is necessary, including the very subtle fine-tuning. We have a proof of concept. Our technology works, and among many other things, it is uniquely positioned to address the challenges of immuno-oncology in the fight against cancer. Viruses are a new tool in this fight, and what we are doing is turning it from a blunt apparatus to a highly accurate instrument. Thank you.
For the presentation, I have 2 questions. One is, can you elaborate more on the proof of concept that you have explained? What viral changes have you induced and what are they doing? And second question is there also potential application of your software platform in the area of infectious diseases?
Sure. So I'll start with the first question. So yes, our proof of concept consists of actually designing and building and testing different kind of viruses. One of them is dengue. This was just as a proof of concept. Zika virus, and another virus for actually a veterinary disease. One of the things is that it was important for us that all of our actually technology, all of our algorithms are completely generic, meaning that if a company, let's say, wants me to, and that happened, actually 2 companies from actually the veterinary field wanted to say, hey, we have just this virus, and could you please develop us a vaccine? We now don't need to start and learning all the proteins and what they do, and then we can just look at the virus from a different point of view, and without changing the proteins, everything is completely generic, attenuate the virus in the specific host. So this is what we did. We showed it on a preclinical studies in animals. Other things we are, well, I'll continue with the second question until we have time. Applications for infectious disease, of course. So as I just mentioned, although cancer is like my, personally, my biggest, but definitely infectious disease vaccines, this is definitely relevant. From a business point of view, we're starting with veterinary disease. This to be fast to market, proving our concept over and over again, and also because viruses from the animals are actually sometimes the basis, and we are learning from them and can be utilized then as oncolytic viruses. And we also find discovered some new novel oncolytic viruses which we patent.
Thank you. Can you elaborate on the manufacturing path, but also the regulatory path that you plan to pursue?
Sure. Again, this is regulatory is the intended use that we are. So obviously for a veterinary field, it's completely different than for cancer.
For human application.
So for human application. So, but I'll answer this quickly. For the veterinary vaccines, it's actually we're working with companies, veterinary, that we're just bringing them, and they are actually moving it. We don't wanna waste the energy on that process. We're delivering them the candidates, and they're going through the regulation for us, for human, and because this is something that in the end we would like to at least start phase 1 and efficacy trials on humans. So we're going obviously for indications that are cancers, that have no current alternative today. So obviously the FDA is much more liberate with going with engineered viruses, and have already recognized, TVEC as an oncolytic virus that was approved. So of course, yes, it's biologics. It's long, but it's possible.
So it's non-personalized. I'm not sure if I got that actually.
It's non-personalized. This is even taking us further along, but but it's really looking at the virus and making sure that it destroys and it's as much as the tumors that we're going afterwards, and be efficacious as possible and safe. Personalized is actually 2 steps. I mean, we're talking about timelines to market. We can do it, but the first, and also in terms of costs, and want to be able to deliver those candidates to patients. So.